We do need to get to diffusion of water a little bit. This is called osmosis, and it is a chronic example of, well, something that trips up medical students. Most of you probably figure we put it in here just to make it difficult for you. And in fact, we did not do that. It is actually very medically relevant, okay? Osmosis is the transport of water, okay? In almost all cell membranes, are very water permeant, usually because there are a lot of aquaporins, which are water channels in those membranes. There are exceptions, like in the kidney, but most membranes are very highly permeable to water. So you have, so it's very important to understand the characteristics of water transport, because we have a situation where if you have an imbalance of water, if we have more on the outside of the cell than the inside of the cell, then the water flows down its gradient and the cell swells. And as you guys probably can figure, cell swelling is often not good. And you'll get side effects, and eventually if the cell swells enough, it'll burst, and that obviously is not good. So cells, generally speaking, can't stand water gradients, because if there's a gradient for water, that means that cell is going to swell, and if it's a maintained gradient, that means it's going to burst, okay? And that's why this is fairly important. You pretty much have to have the same concentration of water on both sides of the membrane, okay? You can't have water diffusing across the membrane. So water concentration is generally measured in terms of dissolved solute particles, okay? So here I have two beakers with 1 liter of water, and the light-colored molecules are water, and the black molecules are space-eating solutes, okay? Now each of these beakers has exactly 1 liter in it, but if you count the number of water molecules in that 1 liter, you can see that you have more water molecules in A than you do in B, because within that same liter, you have space taken up by solutes in this beaker, okay? So you could literally count the molecules, and you could say, I have fewer water molecules here in 1 liter than I have here in 1 liter. So the more molecules I have, the less water I have. So high osmolarity means low water concentration. If water diffuses from high concentration to low concentration, that means it's going to diffuse from low osmolarity to high osmolarity, okay? Because high osmolarity means low water concentration, okay? So again, animal cells can't stand concentration gradients, okay? So the osmolarity is the concentration of particles other than water in solution. So if I have 150 millimolar glucose, we say that we have 150 milliosmoles per liter in that solution, okay? Because a milliosmol is obviously a millimolar of whatever substance you're talking about, okay? So that seems relatively simple. Unfortunately, it gets more complicated. If I have 100 millimolar sodium chloride, I don't have 150 milliosmoles per liter. Because as you guys remember way back from inorganic chemistry, nightmare comes back, it haunts you. Sodium and chloride separate in solution, and they become separate ions. And because they become separate ions, they're separate particles. So 150 millimolar sodium chloride is actually 300 milliosmoles per liter, because each molecule of sodium chloride becomes 2 particles in solution, okay? If I have 150 millimolar calcium chloride, how many milliosmoles per liter do I have? 450. Good work. Because we have 1 particle of calcium and 2 particles of chloride per molecule of calcium chloride. Okay? Good. We use the term osmolarity. Isoosmotic means that your osmolarity is exactly the same as a reference solution, okay? So if I have a reference solution, beaker 1, and I have beaker 2, my osmolarity is relative to beaker 1, and beaker 2 is relative to beaker 1. If it's isoosmotic, that means they have the same osmolarity, okay? If it's hypoosmotic, that means the osmolarity is less in beaker 2 than our reference beaker 1. And if it's hyperosmotic, the concentration in beaker 2 is higher, the osmolarity is higher than our reference beaker 1. A lot of the times, physiologically, our reference is the inside of a cell, and our solution is the extracellular solution, okay? Or more likely, it's a bag of solution that you have in your hand. And knowing what the osmolarity is inside the cell, you can tell me whether it's isoosmotic or not before you put it in the patient and kill them, okay? So bear in mind that that is a physical property of the solution. You measure the number of particles, and relative to a reference solution, it's either isoosmotic or it's not, okay? Tonicity is different. It is a biological term, and it is different. Tonicity tells you whether a cell is going to swell if you drop it into that solution or not. And it is not the same, okay? So a isotonic solution means I can drop a cell into that solution, and the cell is not going to change volume. There'll be no diffusion of water. Eventually, at steady state, the osmolarity is exactly the same inside the cell as it is outside the cell in the beaker, okay? Hypotonic solutions cause cells to swell. Hypertonic cause them to shrink, okay? Again, note that this is a biological property of the solution. You must predict what will happen to the cell if you add that solution, or if you add the cell to that solution in the simplest case. In the most complicated case, if you put it into a patient, what's going to happen to the red blood cells, okay?